Thank you so much for joining us. My name is Tom Lynch. I'm the President and Director and the Raisbeck Endowed Chair here at the Fred Hutch. I want to let everybody know to mark your calendar for next month's Science Says, which will be held on November 30th. Trevor Noah, yes, the same Trevor Noah from television, the host of The Daily Show, will talk with some of our fearless scientists about tackling cancer and COVID-19. We look forward to that engagement tremendously, and I just encourage you to all watch your inboxes so that we can make sure that you know the timing of this event. So, as you all know, the other key thing I would say, I notice we have our first question in the inbox. Please ask your questions. The questions are the best part of these, and there is no question that is too difficult or too easy. Please put them all in there, and we'll do our very best to answer them. Some of them we're not going to be able to answer, but as many as you can would be fantastic. So today, as you know, the Fred Hutch is famous for blood cancer research and for breakthroughs. And you know that, for example, bone marrow transplant started here, and we were one of the true homes of cellular therapy for learning how to engineer T cells to be able to fight cancers. And this has been something which has made a huge difference in patients with leukemia and lymphoma. So this afternoon, we're going to be focusing on solid tumors. And one of the big questions I get asked is, what's the difference between a solid tumor and a liquid tumor? The way we think about it is that solid tumors start in solid organs like the breast, organs like the lung, the stomach, the pancreas, the prostate. Those are examples of solid tumors, whereas liquid tumors start in the blood, the bone marrow, and lymph nodes. So one of the things we've seen, and you're going to see this today in several good examples, we're translating our expertise in immunotherapy to take on these cancers and to advance other strategies. Personally, I'm a solid tumor person. I've spent my life treating and studying lung cancer, and that's the disease I know best at this point. And it's gratifying to see what kind of progress you've made in lung cancer over the past 3 decades, I would say. And it's really due to a couple things. Big part of it is reduction in smoking. We have cut the rate of cigarette smoking in the United States dramatically to now we're at the point of about 16 to 17% of adults in the United States smoke cigarettes, which is a tremendous improvement. When I started my practice, it was 35%, and it used to be as high as 55% in the United States. And so, and despite this progress, lung cancer is still the leading cause of cancer deaths in the United States, followed by colon, pancreas, breast, prostate, and again, they tend to be solid tumors. And this is really important for us to make progress because so many of you on this call, one of the reasons that you support the Fred Hutch and are interested in our work is because of our ability to tackle difficult problems and difficult cancers. I also want to emphasize that it's not just smoking cessation and prevention that have made a difference in lung cancer outcome, but we actually are making differences in treatment. Treatments against a marker called EGFR or a marker called ALK have made a huge difference, and immunotherapies have made a difference in lung cancer. We're going to hear today from some of the best Fred Hutch researchers in lung cancer, in stomach cancer, in breast cancer, as we talk about potential advances in this area. So I want to make a quick note before we start. I am very fortunate to hold an endowed chair at the Fred Hutch. The chair was endowed by James and Sherry Raisbeck. I also want to point out that 2 of our panelists today, Dr. Nancy Davidson and Dr. Nina Salama, also hold Raisbeck chairs. I also want to say that James Raisbeck passed away since our last Science Says. James was an iconic figure in the aviation industry, and with Sherry, a philanthropist whose generosity had a huge impact at the Fred Hutch and throughout Puget Sound. He leaves an extraordinary legacy, and he'll be missed. With that, let's introduce our researchers. First to join is Dr. Nancy Davidson. Dr. Davidson is a physician scientist and a giant in the world of breast cancer. She's the Senior Vice President and Director of our Clinical Research Division, and she's the President and Director of the Seattle Cancer Care Alliance. As I mentioned, Nancy holds the Raisbeck Endowed Chair for Collaborative Research, and the collaborative piece of this is actually important since really one of the things that Nancy has done in her career is bridge that connection between patient care and cancer research. And I think it's a very important part of what we're trying to do here at the Fred Hutch. Her own research on the role of hormones in breast cancer has had a major impact in developing new standards of care. Our ability to treat women with advanced breast cancer has advanced dramatically over the past 3 decades. Nancy, welcome. It's great to have you here. Great to have you talking to the people who are closest supporters of the Fred Hutch. So the first question I'll ask you is, what's happening in breast cancer? What are you most excited about? We just had the ESMO meeting, which is the European equivalent of ASCO. What coming out of ESMO and heading into San Antonio, what are you most excited about in breast cancer?
Tom, there are a lot of areas I could focus on, but I'm going to just focus right now on the therapeutic aspects. One of the big insights for us over the last couple of decades has been the subsetting of breast cancer. We put the breast cancers now into 3 different categories: the hormone responsive ones, the ones that are HER2 responsive, and those that don't respond either to anti-HER2 or hormone therapies. Now, the therapy was pretty standard for a while: chemotherapy, hormone therapy. But now with this understanding, we can begin to think about how we can augment our approaches in each of them. So for example, Tom, right now in the hormone responsive breast cancers, we're looking at some signaling agents called the CDK4/6 inhibitors. We're looking at PIK3CA inhibitors. So we can begin to think about how to add on to our hormone therapies. In the triple negative breast cancers, the hardest ones, because they don't have a lot of targets, we now are seeing new targeted therapies. We're seeing in some cases PARP inhibitors if they have abnormalities of DNA repair. We're seeing that sometimes we can use something called sacituzumab, a new immunotherapy. And we're realizing that triple negative breast cancers are sometimes susceptible to the other immunotherapies, the checkpoint inhibitors. And finally, anti-HER2. We've got 5, I think more than 5, specific different anti-HER2 targeted agents now, which is such a change from 20 years ago when these came on board.
And so, Nancy, can you give us a sense of what the spectrum of breast cancer looks like? So how many cases are there in the United States? What percentage of women present, or I shouldn't say women, what percentage of people present with breast cancer that's localized versus distant? And how do you think of metastatic breast cancer compared to all breast cancer?
Thankfully for us, metastatic breast cancer is a subset of breast cancer, but an important one. 40,000 men and women die for breast cancer each year. Some because they come in with metastatic breast cancer, but many because they develop it after seemingly successful treatment for early breast cancer. That means that about 250,000 new diagnoses, most of which are going to be with early breast cancer, thanks to the availability of screening. Breast cancer is one of those poster children for screening, and that means that most people come in with early stage disease that can be managed with surgery, radiotherapy, and some sort of systemic therapy, and then they go on and never ever really suffer from their disease again. But those metastatic patients, big area for us because we understand these targets now, and we can think about how to use some of these new agents. And once they're worked out there, then we can think about whether we should strategically deploy them in certain circumstances in early breast cancer.
I think in your career, one of the ways that you've made such a huge impact in cure is you've developed drugs that work for patients with metastatic disease, perhaps make them live longer. And then when you've applied those to women with early stage breast cancer, we're actually there increasing the cure rate, which is actually a remarkable thing.
Absolutely right. I think we're seeing both. Sometimes these approaches, and this is a team sport, as you know well, Tom. Sometimes these approaches are helping women with metastatic breast cancer to live longer and without much in the way of side effects. And those are the ones that we're very excited about because we could think about taking those approaches and, as you say, moving them back in a selective fashion to earlier stages of breast cancer to try to hope that individuals with early breast cancer will never progress to metastatic breast cancer.
And so, Nancy, when a woman presents with a breast lump or an early stage breast cancer, that's when the initial molecular profiling is done. And what do you profile for a woman who presents with early stage breast cancer?
Yeah, we are pretty routinely looking at the size of the tumor, the lymph nodes, and so forth. But from a biological perspective, we look at the expression of the estrogen receptor protein, the progesterone receptor protein, because those are proteins that speak to hormone responsiveness. We look at the expression of the HER2 protein, because those breast cancers might be responsive to those HER2 agents. And then we look at the absence of those, the triple negative breast cancers. Increasingly, though, Tom, we're trying to get beyond this one gene, very small subset. And so breast cancer is one of those areas where multi-gene panels are also being used quite extensively. In particular, in early stage hormone receptor positive breast cancer, where we're trying to decide whether chemotherapy should be used in addition to the hormone therapy, we often turn to these multi-gene panels, things like Oncotype or MammaPrint, to try to help us figure out which are the tumors that can be successfully treated with hormone therapy alone, and which would those patients be helped by the addition of chemotherapy. So that's kind of still primitive, and I think we're really hoping that with time we're going to be able to develop even more effective ways of trying to help to sort out response and resistance early.
And so if a woman is diagnosed initially with breast cancer that is ER positive, PR positive, HER2 positive, does it ever change when the woman has metastatic disease? And if they present with triple negative disease, do they ever develop hormone positive or HER2 positive when they have metastatic disease? What happens in between those two times?
Well, you're right. There are so many steps that tumors have to go through to go from early to more advanced cancers. And one of the things we've learned is if someone develops an advanced cancer, it's not enough to rely on the markers from the original biopsy. You do need to do what you just said, which is to recharacterize those markers on the recurrent tumor in order to find out if there has been a change in the behavior of the breast cancer so that you can try to hone the selection of those therapies. With regard to your second question, Tom, it's reasonably common for a hormone receptor or responsive breast cancer to become unresponsive or HER2 positive. It's pretty unusual for one of those triple negative breast cancers to change their stripes. Right. They don't usually swap out. They seem to start that way and stay there for the duration of the disease. But this is what we're learning because of science that can be done. The techniques that are now available to be able to, first of all, do safe biopsies. Second, sometimes we're looking to see whether we can look at this in circulating markers. This is being studied right now. And we realize how important it is to characterize the disease that's in front of us right now, as opposed to relying on the profile that may have come from several years previously.
Nancy, thank you very much. We're now going to talk with Dr. McGarry Houghton. So McGarry leads our lung cancer research program. He's a professor in the clinical research division and a professor in the human biology division here at the Fred Hutch. He's a doctor and a scientist who studies white cells, which are part of our immune system's frontline response to infections. He's also a pulmonary doctor who studies the lungs and thus has a really good understanding of lung biology. His research has challenged convention by showing that tumors can sometimes co-opt neutrophils for their own ends. He's also the principal investigator of our SPORE grant here at the Fred Hutch, and he also leads our lung cancer program in the cancer consortia that we share with the University of Washington and Seattle Children's. McGarry, terrific to have you. And I guess I'm going to start off by saying there has been progress in lung cancer, but it's still the leading cause of cancer death. And I have to tell you, when we discovered EGFR mutations and other people identified ALK and other people identified ROS1, among other subtypes of lung cancer, we really thought that we'd be seeing lung cancer become a chronic disease. And yes, for a very small percentage of patients, that's true. Why aren't we curing more lung cancers now?
Yeah, I think there are two major reasons for that. I mean, one, I think it really speaks to how aggressive and difficult of a cancer lung cancer is to treat. So, if you look historically at traditional chemotherapies and radiotherapies used for many cancers, historically, there is some good effectiveness of those therapies in many cancer types, but not really in lung cancer much at all. So it took these more sophisticated therapies, like you're mentioning, the EGFR antagonists, other targeted therapies, and now the new immune-based therapies to really make a dent in that at all. But it's still a minority of patients that benefit from those. It's about 20% of patients benefit from immune-based therapies. And if you add up all the targeted therapies, that's still maybe somewhere around 20%. So I don't think these more sophisticated, effective therapies are reaching the majority of patients. And then I think the other thing that really is a problem is this idea that we diagnose most lung cancers still at a late stage. So if we diagnose someone stage 1, they can have a surgical resection. Their 5-year survival rates are very, very good, maybe 70%. If they're diagnosed at stage 4, as you know, that's a very different outcome, and survival rates are much poorer. So I think we need more effective therapies to reach more cancer patients, and we need earlier diagnoses.
So McGarry, we just heard from Dr. Davidson, who talked about how early detection is so important in breast cancer. Now, you're a pulmonologist, so you know what the pulmonary doctors are like. We do have evidence that screening for lung cancer can also be effective. And yet we don't do nearly as much screening for lung cancer as we do for breast cancer. Tell me a little bit about that and what some of your colleagues who are pulmonary doctors, how they approach the screening question.
Yeah. So for people who don't know, and unfortunately I think a lot of people don't know, that Medicare and insurance companies now pay for CT scan screening for lung cancer for people age 50 and older and with a robust smoking history. Unfortunately, only 4% of the people that are eligible in the United States to have the screening actually receive it. And I think you can blame lung doctors for a lot of this.
Well, I'm not blaming lung doctors.
No, but you should. You should. So I think historically, CT scans, they find lung nodules. They don't find cancer. And then a lung doctor has to tell you if it's a benign or a malignant nodule. And we're not very good at that. That has always been a problem. And it didn't really matter before, but now with screening, we are overwhelmed with consultations for pulmonary nodules and to determine whether or not they are benign or malignant. So there's really two things. One, despite the fact that very few people get screened, we still see lots and lots of these patients because this is such a common problem. 25% of the population has a lung nodule if you put everybody in the scanner. So these are extremely common. The minority of those are going to be malignant. So we cannot just resect these surgically. We cannot biopsy everybody, right? There are going to be too many harms and too many bad outcomes. So we are trying to improve our ability to look at a CT scan image and know what is there. What are the contour features? Can we look at those more sophisticated with the mathematical data and not just what your eye sees? And then can we add some blood markers? So now we're looking in the plasma. Is there some protein that is apparently expressed by lung cancer? Has your immune system developed a tumor-specific antibody that you can detect in the blood system? So there are efforts here and in other places to combine blood, imaging, patient history, and try and come up with a much better risk prediction algorithm to distinguish the nodules.
And so, McGarry, one of the areas that you've made huge contributions in and that your group here at the Fred Hutch is making big strides in is understanding the immune system and how it can be used to improve lung cancer treatment. Can you give us a sense of where you think some of the most exciting opportunities are there?
Yeah, I think if you go back and say, immune checkpoint inhibitors, the PD-1 antibodies introduced 10 years ago, that was a leap forward in lung cancer. And that was a really big deal and has changed the way that we treat many patients. I think the 10 years since then has been trying to figure out exactly why those drugs worked when they worked and exactly why they didn't work when they didn't work in patients. And so the basic scientific studies that have taken place over the past decade have really taught us why lymphocytes fail to get to the cancer cell. So the CD8 T cell, that is the thing that actually kills cancer cells, it has to get right up next to a cancer cell to kill it. So we are learning, why can't it get there? Why can't it traffic to the right spot? And then when it gets there, sometimes it's not functioning properly and it's not turned on. And we're learning exactly why it's not turned on. I think we're on the precipice of another leap forward. When we really understand how to take that information to get the T cells in the right spot and get them to function properly at the right time, then I think we'll see another big jump in the efficacy of those drugs. And I also think you'll see that information and knowledge can be utilized in other immune-based therapies like a CAR T cell, right? So once we know how to get that cell into the right spot, those therapies become more effective as well.
McGarry, thank you. And I know we're going to have more questions on lung cancer when we talk during the Q&A section. I'd like now to introduce Dr. Nina Salama. Nina is a microbiologist and a leading expert on Helicobacter pylori, a common bacterium that's strongly linked to stomach cancers and some lymphomas. Her team has made fundamental discoveries about the shape and function of Helicobacter pylori, which is helping explain how this drives cancer. And it's really important for people to recognize that we're not going to be able to improve the treatment of cancer if we don't understand cancer better. And the fundamental work that Dr. Salama is doing in her laboratory is making a huge difference in giving us that understanding of that. Nina also holds the Dr. Penny E. Peterson Memorial Chair for Lymphoma Research, which was generously endowed by James and Sherry Raisbeck. Nina, welcome, and thank you so much for joining us today. So I guess the first question is, you study bacteria. What should people know about the role that bacteria play in cancer development? Because I can tell you, when I trained in oncology, we were never taught about any relationship between bacteria and cancer. We were taught about viruses, but not bacteria.
Well, thank you very much, Tom. So I think you bring up a really important point, that a lot of people don't realize that about 15 to some people even think up to 20% of cancers are caused by an infectious agent. And when I say infectious agent, that could be a virus, a bacteria, a parasite. The more famous infectious causes of cancer are viruses. So human papillomavirus for cervical cancer, hepatitis viruses for liver cancer. But it turns out that on an agent basis, the most common infectious cancer is actually Helicobacter pylori, the bacteria that I work on, which causes stomach cancer. So it has a huge burden. It turns out stomach cancer is the 5th most common cancer worldwide and the 4th most common cause of cancer death. There's a very wide regional patchiness to gastric cancer, so it is not as common in the United States, but still we have about 26,000 cases and 11,000 deaths a year in the US from stomach cancer. So it's a really important problem. Now, one of the things that's hard to think about is, so for a virus causing cancer, we know that viruses invade cells and they could, express an oncogene, for example, and drive a cancer. But while some bacteria can invade cells, it turns out Helicobacter pylori doesn't. And so the really big million-dollar question is, how does it cause cancer? And interestingly, one of the major hypotheses is that it relates to how it skews the immune system. And we just, heard from McGarry about how important the immune system is in affecting both cancer etiology and cancer treatments. So I think in terms of impact for patients, first, anytime you've got an infectious cancer, targeting that agent is a promising way to prevent and even possibly treat cancer. But then also in terms of thinking about therapies that exploit the immune response, if you've got a microbe that's manipulating the immune response, that gives you another handle both to understand the etiology and perhaps treatment.
And so Nina, we have 200 people on the call on the program today who are supporters of the Fred Hutch, and I'm sure many of them are saying, well, how do I prevent myself from getting H. pylori? Is this something that you can prevent yourself from getting? Is it something that we are all exposed to? Tell us a little bit about that.
Yeah, so that's a really good question, which is still not very well understood. So Helicobacter pylori is actually considered a part of the microbiome. So bacterial pathogens, some of them like Vibrio cholera, for example, normally live in the water and you only ingest them accidentally and then they cause havoc and go away. But then there's other bacteria that are actually part of our microbiome and then only in certain situations can cause disease. And Helicobacter pylori is actually one of those. So Helicobacter pylori lives exclusively in the stomach, and it's actually transmitted within families, and the infection is acquired very early in life. And we actually don't know exactly the precise route of transmission, nor really how to prevent it. I have to be honest.
And so, Nina, as you point out, lymphomas, solid tumors, why would a bacteria cause a lymphoma one time and a solid tumor another time?
That is another interesting question that we don't fully know the answer to. So the most common cancer associated with Helicobacter pylori is adenocarcinoma, so a cancer that arises from the tissues of the functional tissues of the stomach. But in rare cases, Helicobacter can also induce a lymphoma. And it's thought that that is part of the fact that normally we don't have much of an immune response in the stomach. But when you have H. pylori infection, you get this influx of immune cells. And in some cases, those actually can become either a B cell or a T cell lymphoma. One of the things that's fascinating about that particular lymphoma is if you treat with antibiotics to get rid of the antigen driver, you can actually cause regression of the lymphoma, even at quite late stages. So pathologists find this tumor very fascinating.
And Nina, one of the things we're doing at the Hutch is we're recruiting scientists to come here who study the microbiome and are really interested in the bacteria that live normally in our bodies and what role they may have in causing cancer. Can you talk a little about the collaborations you formed at the Hutch and how this is becoming a big area of focus for us?
Yeah, I'll have to say that, back even from when I was hired 20 years ago, the collaborative nature of Fred Hutch was one of the big attractive points for me to come here. And I've had collaborations throughout my time here, but I'm actually super excited about a new collaboration that we just got a small grant from the Gastric Cancer Foundation for. And this is a collaboration between my lab and Susan Bullman's lab in the Human Biology Division and Megan Koch's lab. And so what the focus of this grant is on is actually to look at the possibility that Helicobacter pylori collaborates with another bacterium that normally lives in the mouth, Fusobacterium nucleatum, but which Susan has shown can impact colorectal cancer all the way down at the other end of the GI tract, that it and Helicobacter might collaborate together. And we think that that this is important because we think that some people have only Helicobacter and some may only have Fuso, and they would have very different immune responses. And that's where Megan comes in, Megan Koch, who studies in intestinal immunology more often in the context of maternal protection via antibodies from disease, but using some of the tools that she's developed to study the infection response in the GI tract. We're going to take her up to the stomach. So that's an example where we're pulling people with different expertise from different sites and getting them focused on the stomach.
So thank you, Nina, very much. We're going to be back with you for questions in just a second. But we would be remiss if we didn't have a section on COVID. And I'm delighted to introduce Dr. Josh Schiffer. Josh is an associate professor in the Vaccine and Infectious Disease Division. He's also an associate professor in the Clinical Research Division. He's an infectious disease doctor and a researcher who, as you could imagine, has been remarkably busy since the beginning of this pandemic. His team uses mathematical modeling to understand all the variables that may be involved in the spread of COVID. He was among the first to recognize the super spreader phenomena. He is a very strong advocate of masking and other measures to prevent spread, and he's one of the many researchers who are leading trials here at our COVID-19 Clinical Research Center. So I guess the first question for you, Josh, is everyone's concerned about boosters. Tell us about boosting adults versus vaccinating kids for the first time. We heard today in the New York Times and in the Wall Street Journal and in the Seattle Times, all three reported that Pfizer will be asking for, and Moderna, both of them will be asking for approval in children. Which is going to be a quicker way to getting us there? Boosting adults, vaccinating kids?
Well, thanks for having me today, Tom. The question of boosting is complicated, and I think that it's different if you answer it at the individual level rather than at the population level. So, many friends and colleagues have come to me and asked if they should get the booster, and my answer is, if you fit within the guidelines, yes, you should. But as far as ending the pandemic, boosting is far less consequential than vaccinating the remaining unvaccinated portion of the population. So we just submitted a preprint that I worked on with Dobromir Dimitrov, Mia Moore, Chloe Bracis, Dave Swan, and others, where we really looked at the question of vaccinating children. And what we found was that if we can vaccinate children soon, that there is the potential to decrease hospitalizations and the chance that we would need to lock down at some level due to a surge in hospitalizations significantly, and to reduce hospitalizations both among kids and the general population. But the other thing that stuck out is the need to really focus on the remaining unvaccinated people. As much of a challenge as this is, within King County, the model projected that if we were to vaccinate 10% more people, so that's about half of the remaining unvaccinated people in King County, among people who are eligible for the vaccine already, so adults, that we could really stave off any significant further wave of infection. And so the strategy really remains the same, which is to vaccinate as many people as possible, and that includes both children, but also people who have remained hesitant to get the vaccine up to this point.
And so, Josh, like you, I get a lot of questions from people about vaccine safety. We saw just this week that there was a death in King County from the J&J vaccine as a side effect from a bleeding issue in a younger woman. And yet we believe that the overall safety of vaccines justifies their use. So a couple of questions. Can you put relative risk of side effects in perspective for people? Now, I assume the vast majority of people on this call are going to be vaccinated, but there'll be some who aren't. And more importantly, all of us talk to people who are not vaccinated. I, even as a president of the Hutch, have family members who've chosen not to be vaccinated, as hard as that may be. So the question is, what do you advise us for how to talk to people who aren't vaccinated?
Yeah, I mean, it's frankly very challenging for me. I need to be honest about that. It's challenging for me because I am so saddened and heartbroken by the fact that we continue to lose hundreds of lives unnecessarily. And there's no getting around the fact that the unvaccinated are contributing to this. That's just the scientific fact. But I think in general, we need to try to put those frustrations aside and meet people where they are and listen. And, you know, I've probably spoken with maybe upwards of 10 friends of friends who were unvaccinated to try to change their minds and get them vaccinated. And I certainly have not succeeded with everyone, but the common denominator seems to be time. You just have to put in a lot of time to the conversation and really understand what people's misgivings are and patiently listen to them and walk them through it.
And so, Josh, as people on this call know, really because of the generosity of people who support the Hutch, we were able to establish and open a COVID-19 clinical research center here at the Fred Hutch, where clinical trials are done. And one of the drugs that was looked at in that clinical trial is a drug made by Merck called molnupiravir, which is an oral agent which appears to reduce the risk of hospitalization and death by almost 50%. Tell us about this drug. Tell us about what role you think it will play. How close do you think this is to being available for people?
Yeah, thanks, Tom. If it's okay, I'd like to touch on two themes here. The first is the drug. The drug, it's marvelous. The reason it is such a breakthrough is that it's an oral agent. And while the monoclonals were really a tremendous scientific advance, a lot of us were very concerned from the beginning about implementation, and that unfortunately turned out to be true, that only a vast minority of people who are at risk of severe COVID who get infected end up receiving a monoclonal. And so this has the potential to be a game changer. You can imagine a situation where somebody gets diagnosed, gets contacted, and has the drug in their system within 12 hours of receiving a diagnosis. And that's the theme I really want to harp on, is that the COVID CRC was built on a scientific principle. And that principle was that we knew both from mathematical modeling, but also from clinical precedent with influenza and Ebola and herpes viruses and HIV, that treating early is always better when using a directly acting antiviral. And it's worth noting that the CRC has contributed in a major way to 4 studies showing 2 monoclonals, Vir and Regeneron, but also the molnupiravir and most recently remdesivir, studies which unequivocally showed reductions in hospitalization rates and unequivocally showed far higher benefit in treating people early than in people who received the same agents in the hospital. So it's really just an incredible story that the Hutch was able to put this together so quickly and then recruit so many participants in a city where the incidence of cases was actually lower than many other sites so effectively and contribute to these 4 incredibly important trials.
It's really remarkable, Josh. And I guess the last question I'll ask you before we bring everyone back is, do you worry a little bit that the availability of an active oral treatment might cause some people to say, I'm not going to get vaxxed. I'm just going to wait and just take the pill if I need something?
Yes, I do. I mean, we've now had people who aren't getting vaccinated because the belief of the availability of agents that are effective that are not effective. That's now happened 2 or 3 times. And unfortunately, that has taken on a partisan divide, which is like inexplicable to me and incredibly sad. But, you know, the reality is that we need every tool in our armamentarium. One of the points our models made is that none of the vaccination stories matters if we don't continue to have masking and relative social distancing in schools. So until this thing is over, we need to use every tool that we have because none of them are perfect. But when you bring them all together, we can reach a situation where cases stop spreading and the few cases that we have are not lethal. So yeah, I think the messaging of the new drug will need to be really done precisely and carefully so that we continue to vaccinate as many people as we can.
Great. Josh, thank you. If we can now bring everyone back together, if Josh stays on, bring back Nancy and McGarry and Nina. Thank you all for coming back. So the first question I'm going to ask will be to Dr. Davidson. So Nancy, you and I have both been at more places than just the Hutch. You were at the University of Pittsburgh and at Johns Hopkins and at Harvard before all of that. And I guess the question that people always ask is, well, I'm interested in supporting cancer research. I want to support breast cancer or colon cancer or lung cancer. Do you think that it matters what type of cancer people support, what kind of research, or do you think that supporting cancer research in general helps a lot of different cancer patients and a lot of different cancers? Tell me about how you think about how we use the funds we raise in cancer research.
I think that both approaches are really valid, Tom. You know, a lot of our philanthropy is based on sort of disease-specific support, and I would never want to discourage that if that's what the donor is really intent on. But I want to pick up on your theme that, you know, sometimes you can support one area and find out it has a lot of impact across multiple areas. And here I'm going to shout out the research that's gone on in the field of DNA repair and BRCA susceptibility. You know, we're at the city where Mary Claire King lives. She was the person that helped to identify BRCA1 and subsequently BRCA2 and a whole family of DNA repair genes. And, you know, when you have mutations that you inherit in those genes, you're more prone, we learned originally, to breast cancer. And then we learned to ovarian cancer. And then we learned through research that was done here that that's also true for men with metastatic prostate cancer. So I would suggest that people who went out to support research in DNA repair and thought in these susceptibility genes, who thought that they were helping breast cancer research, they did a great thing. But their work, you know, their support had enormous implications across other solid tumors. Breast, like we talked about, ovarian, this prostate work led by people like Heather Chang and Pete Nelson here, and pancreatic cancer. So I think that you can't underestimate the importance of supporting fundamental research and how it might play, you know, have dividends, pay dividends in ways you cannot possibly imagine.
Thank you, Nancy. Nina, there's a question that just came in, which is gastric cancer is a big cause of cancer death in East Asia. And in other parts of the world, it's a much more common cause of cancer death than it is in the United States. Why is that? Are the microbiomes different? Are there other reasons that can explain that? And how what do we know about global gastric cancer?
So I would, in terms of what are the differences, the microbiome, the H. pylori strains, the other exposures, I would say yes to all of that. We've been involved in some studies looking at strains from China and Japan, and there definitely are specific differences in the H. pylori genomes that probably account for part of the answer, but not all of the answer. And one of the kind of regional differences that I find most fascinating is that there's a stripe of really high gastric cancer that goes down Central and South America, essentially along the Andes. And so in one country of like Colombia or Costa Rica, you have different gastric cancer rates in people living in the mountains versus the coast. And some of that difference is actually thought to be relating to a mismatch between the host genetics and actually the Helicobacter pylori strain. So it's a very complex interaction that explains some of these differences. And so I had mentioned that there's this sort of patchiness before, and we don't fully understand why is there higher gastric cancer in Eastern Europe rather than Western Europe, for example. And even here within the United States, there's a lot of health disparities. For example, African American men have a higher incidence of gastric cancer, and then also worse mortality if they have gastric cancer. So I'm currently involved in a collaboration with Duke University to look at that health disparity.
Okay. Thank you, Nina. So McGarry, question has come in for you. As a pulmonologist, what do we know about the current status of the vaping epidemic? Can you get lung cancer from vaping? As a pulmonologist, have you seen people come in with damage to their lungs from vaping? Is it safe? If you're a smoker, should you think about vaping as a safer way to consume nicotine? What do we know about that, McGarry?
Well, I'll start out by saying, for people that remember the news, and this was before the pandemic, just the summer before, there was an outbreak of fatalities in the Midwest, Wisconsin, Minnesota. That from vaping, these tended to be adolescents, people in their young 20s. Now, that was linked to a specific agent that was a contaminant in those particular products. So for people thinking of that should not be the case anymore. There's some more regulation for that. If you're talking about what vaping does, there's still really not a lot of good research. There's been no sort of preclinical animal model type stuff hardly at all done. But what we do know is that you can generate emphysema in animal models with vaping. And these are complicated things to think of. But what I would say is that your lungs were designed to inhale good quality air and put the oxygen into your bloodstream. That's what they were designed to do. And if you chronically inhale anything that isn't good quality air, whether that's cigarette smoke or wood smoke or vape products, or remember, people that cook with wood smoke fires in South America and Mexico, that's a common cause of emphysema worldwide. So it's the particulates, the little tiny particulate matter, no matter what it's from, it gets deposited in the lung tissue and it causes chronic inflammation. And that chronic inflammation causes lung damage. So I don't think vaping is going to prove to be much better than cigarettes. The cancer question is a little bit more challenging, but there's certainly some evidence that vaping is going to have a cancer signal. But the chronic lung disease signal is going to be a real problem with those problems.
McGarry, thank you. Josh, this next question is for you. And it's from somebody who says, my dad is 78 and is fully vaccinated and also has had COVID. So fully vaxxed and COVID. Does he need a booster? Okay. And when will the Moderna booster be out?
So to the first question, I think that if her father had received the Pfizer vaccine specifically and did so more than 6 months ago, then I think he would be eligible and should probably check in with his physician in regards to timing as to when he was infected with COVID, because there might be some bearing upon when best to receive the vaccine. The Moderna vaccine, I don't know, Tom. I haven't followed the story closely enough as to when that booster will be, first of all, available and second of all, recommended.
But can you comment as well on this concept of what people like to call natural immunity? So the immunity you get from having had COVID compared to the immunity you get from the vaccine. And we've done some work here at the Hutch, which has given us some insight into that.
Yeah, so in general, the natural immunity related to COVID infection, surprisingly, is generally on average less than that associated with the vaccine and more variable. So it's much tougher to predict whether somebody will generate a viable long-term response against the virus if they've been previously infected, though clearly there's on average some level of protection. What's fascinating is that people who receive the vaccine after getting infected appear to have the optimal level of protection among everyone who has been looked at. And so it's all the more reason for somebody who has had COVID before to get vaccinated is that it really does put them in a special category of protection, though, as with everyone else, the story of waning immunity, I think, remains to be told. I think the overall evidence in terms of waning immunity is probably that people will lose protection against getting infected, but that most people will retain protection against severe disease. And this relates to what we're hoping we will start to see soon, which is that the rates of hospitalization will start to dissociate from the rates of cases, and that those rates of hospitalization will become much lower. And that's really, I think, when epidemiologists will start to feel that the pandemic in the U.S. is coming to a close, is when the hospitals are no longer feeling surges of COVID, despite the fact that there are remaining cases in the community.
Great. Thank you, Josh. Well, this next question is not for Nina, who works on the immune system and how it impacts H. pylori, not for McGarry, who works on the immune system and lung cancer, or Josh, who is an immunologist in terms of thinking about how our body responds to viruses, but rather to Nancy. And the question comes in is, tell us about immunotherapies for triple negative breast cancer. And is this an option that can help patients?
Yeah, it is funny that you're right, Tom, that you're coming to the non-immunologist here. But this is an area that we're pretty excited about in breast cancer right now. And right now, I think of immune therapy and triple negative breast cancer is coming sort of in some ways in two different flavors. One is that McGarry touched earlier on the checkpoint inhibitors, these agents that have become useful across a variety of tumor types. And in breast cancer, it does look like the triple negative ones are the most susceptible. The one that seems to be the most effective right now is the agent called pembrolizumab. And right now, the FDA has approved pembrolizumab both for advanced triple negative breast cancer with chemotherapy, but also for what we call neoadjuvant therapy. So sometimes with patients who come with an early stage of triple negative breast cancer, we think that they would be best served by a course of chemotherapy before they go to the operating room for their surgery and their radiotherapy. Neoadjuvant, we call it, or preoperative. Well, a big trial was done that suggested that if you use this pembrolizumab in combination with the chemotherapy preoperatively, and then continue some of that therapy postoperatively, those women do better than those who received only the chemotherapy. So this pembrolizumab was approved by the FDA a couple months ago for use in certain circumstances of early stage triple negative breast cancer when you're going to employ chemotherapy first. It's also approved for metastatic breast cancer in this setting. And I would say that this is an area to watch because, there was another member of this family, something called atezolizumab, which was the first one out of the box. And actually subsequent testing suggested that maybe it wasn't quite as robust as we wanted. And so that has actually fallen off of our radar. So please know this is a really rapidly changing area. I also want to point out one other way of thinking about immunotherapy, Tom, which is that, there antibody therapies that we use, things like trastuzumab or Herceptin for HER2 breast cancer. But there's now a new agent called sacituzumab, which is an antibody that's targeted with a cytotoxic drug. And so it's sort of like a smart bomb where it can try to deliver this cytotoxic drug to tumor cells. And this particular agent has actually come into our repertoire for triple negative breast cancer as well. So even a different way of trying to think about immunotherapy. And if you give me one more sec, CAR T cells, we know all about it in the setting of, we know a lot about it in the setting of blood cancers. We've done a ton of the work here. We should know that there are early stage trials that are going on looking at these CAR T approaches, also in triple negative breast cancers and in some kinds of lung cancers against the target ROR. We're trying really hard to think about the immune system, immune therapies, and how they can be used in breast cancer.
Thank you, Nancy. McGarry, the next question is for you from Janet Sears. It says, thank you for this information, very interesting. My mother was a non-smoker who died at 59 of lung cancer, and I really appreciate your work. My question is, how does one decide to test for lung cancer in someone who doesn't have obvious risk factors? And Janet, I'm sorry for your mom's passing from lung cancer, but this is a really important question, which is, we see a lot of lung cancer in never smokers and non-smokers. And McGarry, how do you advise people should people who are never smokers pursue screening for lung cancer? What's the role of screening and early detection in never smokers? And what do we know about never smoker lung cancer?
Yeah, Tom, that is a question that I get frequently, and I must say is a very difficult question to answer. So if you just start out with the screening guidelines, the guidelines do not include people without smoking. So the guidelines as of today are 20 pack years, meaning a pack a day for 20 years equivalent, and an age of 50. And if you quit not too long ago. There is not a criteria to screen for never smokers. And the reason for that is that, as I mentioned, if you did CT scans on all people, all comers without smoking history, you would find lots of pulmonary nodules. And since the major risk factor is cigarette smoke, the vast majority of pulmonary nodules in non-smokers would be benign. So it'd be a tremendous amount of work and putting patients at risk of repeat radiation procedures through CT scans and other things that wouldn't be of benefit. So I think where things are changing and where the future in this lies is that screening strategies that will go for the whole population that do not rely on a CT scan and would not necessarily rely on a smoking history. And these are looking at what we call risk or fields at risk. So that there's one individual who developed a test that takes a nasal swab actually and looks at the gene expression, and there are profiles that are associated with at-risk fields. So again, if you have a cancer, many of the cells around the cancer are letting you know that they're behaving abnormally, and you can pick up those gene expression signatures. So there certainly are people working in this area. It's some ways away, but it's well known that relying on CT scans for early detection is really not going to work for the general population.
Thanks, McGarry. And Nina, a question about screening. In Asia, some people are screened for gastric cancer. Why don't we screen people for gastric cancer here in the United States?
It's really a frequency of the disease and the fact that gastric cancer rates are so much higher in East Asia. So Japan and Korea have active screening programs, similar to here where you get a colonoscopy, you actually get an upper GI endoscopy to find early cancers, and it's very effective. So they can, if you find cancer early, they can actually resect it with the endoscope. And interestingly, one of the things that they find is that if they also then treat your H. pylori, they can prevent a recurrent cancer.
And this question is for Nancy, and I think it's going to be the last question we're going to have time for. It's from Lee James, and it says, one of my sons died 9 years ago at age 43 of epithelioid hemangioendothelioma. It initiated in his liver. Avastin worked for a while until his kidneys failed. This is a very rare cancer, and at the time we were told that it was not studied extensively. How do we study rare cancers? And Lee, I again express my sympathies to you for the loss of your son. And rare cancers are a major problem because if you look at them collectively, there are a lot of people who do get rare cancers. Nancy, how do we, in a cancer center, how do we do work on things like epithelioid hemangioendothelioma?
I think this goes back to some of the things that you talked about earlier, Tom. First of all, a lot of cancers are becoming rare. Obviously, your son's cancer was really rare. But if you start to do molecular subsetting of some of our seemingly common cancers, you break them down into ever smaller buckets of rare cancers. And so we're having to think about how to do this pretty routinely. Luckily, our pediatric colleagues have been doing this for a very long time. And so I think that there's a lot of science wrapped around it. Certainly one key part is that there's a lot of interest in the molecular characterization or the omic characterization of these cancers to understand pathways. Because as you mentioned earlier, Tom, sometimes these pathways are cross-cutting. So a pathway that might be important in one kind of common cancer could turn out to be also important in this rare cancer. And you might be able to think about how to use those approaches across the molecular continuum. I think this is going to be really the science of the future, is trying to wed the molecular pathways with the histology and trying to figure out how to take that forward.
Nancy, thank you. I want to thank you and all these speakers, Nina, McGarry, Josh, for your participation. I want to thank our ASL interpreters as well for their remarkable work. And I want to thank all of you who helped to fuel and support our work with your support of the Hutch. As you know, federal grants only support about 70% of the cost of doing research here at the Fred Hutch, and we are very dependent upon the help and support of people who understand the mission and support the mission. You can make a gift online. Our URL is on the screen. Just a quick reminder that our next Science Says with Trevor Noah will be on Tuesday, November 30th, which is Giving Tuesday. Please join us. In the meantime, please stay safe and healthy. Thank you.